Cellular survival requires the continuous, perfect regulation of highly reactive molecules. When regulation fails, a microscopic metabolic error rapidly scales into catastrophic systemic tissue destruction. We are going to deconstruct the exact mechanisms behind three specific pathologies: hereditary hemochromatosis, Wilson disease, and acute intermittent porphyria. The first mechanism of failure is transporter breakdown. The body requires certain heavy metals to function, but lacks passive ways to excrete them. If the specialized pumps that push these toxic elements out of our cells fail, we experience systemic overload. The second mechanism is a biosynthetic bottleneck. When a single enzyme in a multi-step molecular pathway is missing, the preceding intermediate molecules pile up, creating a toxic backlog that poisons the surrounding tissue. Mastering differential diagnosis for these conditions means abandoning the rote memorization of symptom checklists. Instead, you have to trace the clinical fallout backwards, logically deducing the exact molecular break that caused it. Iron is a highly reactive transition metal. Because the human body does not possess an active mechanism to excrete it, dietary iron absorption must be tightly controlled at the cellular level to prevent toxicity. The master regulatory brake for systemic iron is a peptide called hepcidin, produced in the liver. Look at this diagram of a duodenal enterocyte. When systemic iron is high, hepcidin travels to the gut and directly binds to the exporter channel ferroportin. It internalizes and dismantles this channel, physically halting the absorption of iron into the blood. This schematic outlines hepatic regulation. The liver senses iron load using a complex of HFE and transferrin receptor 2. As saturation increases, this complex activates a signaling cascade. SMAD proteins act as messengers, translocating to the nucleus to upregulate transcription of the hepcidin gene, HAMP. The primary genetic break in hereditary hemochromatosis is an autosomal recessive mutation in the HFE gene on chromosome 6p21.3. In the vast majority of patients, this is the C282Y mutation. This substitution prevents the mutant HFE protein from forming its sensing complex. Without that sensor, the liver cannot detect transferrin saturation, and serum hepcidin levels remain inappropriately low. Without the hepcidin break, ferroportin channels on duodenal enterocytes remain permanently open. The patient absorbs massive amounts of iron without regulatory limit. This relentless influx quickly saturates all available transferrin. Iron then begins appearing in the plasma loosely bound to albumin. This is non-transferrin-bound iron, or NTBI. NTBI is devoid of its classical carriers and is rapidly taken up by organs. Once inside the cells, it generates destructive free oxygen radicals through the Fenton reaction, slowly tearing apart cellular structures. Over decades, total body iron can exceed 20 grams, leading to a late-stage clinical tetrad: micronodular cirrhosis, destruction of pancreatic beta cells, increased melanin production, and dilated cardiomyopathy. This pattern of parenchymal cell destruction defines primary hemochromatosis. In secondary hemochromatosis, caused by repeated blood transfusions or ineffective erythropoiesis, excess iron accumulates first in the Kupffer cells and macrophages of the spleen and liver, rather than the parenchymal cells themselves. To confirm the primary disease, laboratory studies must show an elevated ferritin level alongside a transferrin saturation exceeding 60%. The definitive pathological proof comes from a liver biopsy. Using a Prussian blue stain, the excess iron will appear as golden yellow hemosiderin granules packed specifically into the periportal zone 1 hepatocytes. The systemic failure of hemochromatosis, the ruined pancreas, the failing heart, the bronze skin, is the direct logical consequence of a single broken HFE sensor failing to deploy the hepcidin brake. Copper is another essential micronutrient that requires a dedicated, tightly controlled processing system in the liver to prevent heavy metal toxicity. This diagram illustrates normal hepatic copper routing. Dietary copper enters the hepatocyte and is delivered to the trans-Golgi network. Here, a P-type ATPase acts as a hub, loading copper onto apoceruloplasmin for blood transport and packaging excess copper for direct excretion into the bile canaliculus. Wilson disease results from an autosomal recessive loss-of-function mutation in the ATP7B gene, most frequently the H1069 mutation. the mutant P-type ATPase completely fails. It can neither load copper into ceruloplasmin nor excrete it into the bile. Toxic copper pools inside the hepatocyte. This triggers free radical formation, visible microscopically as severe steatosis, ballooned hepatocytes, and eventual frank hepatocellular necrosis. As the necrotic hepatocytes rupture, they spill massive quantities of unbound, highly toxic free copper directly into the bloodstream. Unlike hemochromatosis, which takes decades to manifest, Wilson disease presents much earlier. Patients typically develop severe liver disease in childhood, followed by neurological symptoms manifesting in early adulthood. Here is a T1-weighted MRI of a Wilson disease patient. The arrows in panel A highlight bilateral increased signal intensity, demonstrating that the circulating free copper has selectively targeted and destroyed central gray matter nuclei, specifically the putamen and globus pallidus. This basal ganglia destruction creates severe neuropsychiatric manifestations, presenting as dysarthria, resting tremors, rigid dystonic postures, and significant psychiatric shifts. In panel B, we see the definitive clinical hallmark of this systemic copper deposition, the Kayser-Fleischer ring, a golden-brown ring of copper trapped in the desmet membrane of the cornea. The diagnostic lab profile requires careful interpretation. Patients will have a paradoxically low serum ceruloplasmin level. Because the broken Golgi ATPase fails to incorporate copper into apoceruloplasmin, the empty apoprotein is secreted and rapidly degraded. Microscopic evidence is required to confirm the diagnosis. In the brain, this means identifying altered glial cells called Opolsky cells. In the liver, as seen in panel C, a rhodonine stain must be used to highlight the elemental copper as bright red cytoplasmic granules. Wilson disease is a catastrophic Rhoudin error. A single mutation turns the liver from a processor into a toxic reservoir, which eventually ruptures and short-circuits the basal ganglia. The second major paradigm of metabolic failure relies on a biosynthetic bottleneck, best illustrated by the pathway for heme production. Porphyrias are a group of 8 rare disorders. In each one, a specific enzyme deficiency breaks a different link in the multi-step heme assembly line. This flowchart tracks the critical early steps of normal heme synthesis in the liver. The process predictably generates specific intermediate molecules, primarily delta-aminolevulitic acid, or ALA, and porphiobilinogen, or PBG. Acute intermittent porphyria, or AIP, is inherited as an autosomal dominant disorder, distinguishing its genetic pattern from the recessive metal storage diseases we just covered. In AIP, the exact molecular defect is a partial deficiency of the enzyme porphiobilinogen deaminase, or PBGD. This bottleneck halts the assembly line. The biochemical consequence is the active overproduction and massive upstream accumulation of neurotoxic PBG and ALA in the liver. This toxic accumulation triggers discrete acute neuropsychiatric attacks. Patients experience severe abdominal pain, peripheral neuropathy with muscle weakness, and delirium, often precipitated by physiological stressors like fasting or certain drugs. For diagnostic accuracy, note the location of the block. Because the defect in AIP occurs early in the pathway, patients will have absolutely no cutaneous photosensitivity or skin blistering. This sharply contrasts with porphyria cutanea tarda, which blocks a later enzyme. During an acute attack, the diagnostic hallmark is urine that turns reddish-brown due to the high presence of urinary porphyrins and PBG, but critically, tests entirely negative for blood on a standard dipstick. In porphyrias, the clinical phenotype is strictly determined by the exact location of the bottleneck. A block at PBGD creates a toxic flood of early precursors that ravages the nervous and autonomic systems exclusively. Side by side, these disorders reveal core mechanical failures: an iron sensor failure in hemochromatosis, copper excretion failure in Wilson disease, and a heme bottleneck in AIP. Their physical signatures are distinct. Prussian blue isolates iron, rhodonine proves copper, and porphyrin-rich urine confirms an early heme block. Therapeutic interventions directly target these mechanical failures. Phlebotomy is required to manually drain excess iron, chelators strip away toxic copper, and intravenous hemin is administered in AIP to suppress ALA synthase and halt toxic precursor overproduction. Memorizing a fragmented list of clinical signs, like bronze diabetes or psychiatric tremors, is a fragile strategy that falls apart under diagnostic pressure. True clinical intuition is achieved by identifying the exact molecular broken link. Once you understand the mechanical failure, the complex clinical picture maps itself logically.